One.
Good morning. All right, so today's will be kind of short because I just want to finish up this tools part. So we're on electron microscopes, and there's two different types, which I didn't get into a ton: transmission and scanning. Transmission works kind of like a light microscope where it shoots the electrons through it, and you get images similar to this where you can see all the internal organelles and everything that's going on inside. Scanning, on the other hand, bounces electrons off the surface, kind of like a record player. And so you get pictures like this where you can see the surface of whatever you're looking at in great detail. Again, these samples have to be dead because they take a really long time to do. So if you want to look at something alive, you have to use a light microscope. All right, other lab techniques, cell cultures. Cell cultures have come a long way in the last, 70, 80 years. We can grow a group of cells from one, and so then we can better study what that one cell had, whether that one cell had a disease or a unique protein or something we wanted. We can study that very well using cell cultures. Okay. We can also use cell fractionation. That's a fun word. You separate the parts of the cells. And basically, we put them in a blender, something similar to a blender, and then centrifuge it. And so if you've been on a merry-go-round any time in your life, you know the heavier things get pushed to the outside, tries to push you off. And so that's what happens. The heavier parts of the cell get pushed to the outside of the tube, lighter parts in the center, and so you have all the gradations of the different parts of the cell. And then you can study whatever individual part you want. Lastly, safety is always very important in a lab. Hey, safety is number one. You want to prevent diseases if you're working in a lab with some kind of microbe. You want to prevent contamination. If I'm trying to tell if somebody has a disease, I don't want to mix their samples up. I don't want to put bacteria into that sample. So I got to keep everything safe and clean. And in our labs, two biggest things is know what you're doing. Read all the directions first so you know the next steps coming, and wash your hands at the end of our labs. That'll go a long way in the lab safety department. All right, nice and short lecture. Thank you.